Nowadays, immunological interpretation of disease are expanding their territories to almost all of the disease. Even though immune responses arise to overcome the problems of our bodies like infection or allergy, same responses take place in unexpected disease. In the past year, no one took type 2 diabetes as an immune disease. It has been regarded as the metabolic disease caused by insulin resistance. But today, scientists are finding a solution from the immunological point of view in accordance with identifying the roles of inflammation and insulin resistance. Likewise, inflammation became the cardinal cause of fatty streak formation. Chronic low-grade inflammation plays the key roles in the etiology of the atherosclerosis. Cross-talking between smooth muscle cells and the serial cells of blood vessel walls and macrophages by exchanging the signals composed of the chemokine, the pro and the anti-inflammatory proteins and adhesive proteins induces the atherosclerosis through the serial process including monocyte recruitment and adhesion, accelerating the fatty streak formation and reducing the elasticity of blood vessel walls. The focus of this lecture is introducing how the drugs control the part of macrophage to resolve the problems causing the atherosclerosis. In part 1, the action mechanism of drugs on the pro and anti-inflammatory responses resolving chronic low-grade inflammation will be explained. In part 2, the action mechanism of drugs or other materials regulating monocyte recruitment and adhesion will be introduced. Last, in part 3, how the drugs control cholesterol efflux of macrophages in respect to relationship between resolving the inflammation and lipid metabolism will be illustrated.
In part 1, chronic low-grade inflammation and its related genes and proteins will be mentioned with the action mechanism of the drug or single compounds balancing the pro and anti-inflammatory responses of monocyte macrophages in the atherosclerotic patient or animal model. The imbalance of immune responses between the pro and anti-inflammatory action is the etiology of the atherosclerosis. Interleukin-1 beta, interleukin-6, C-reactive protein, and TNF-α, so-called pro-inflammatory proteins, are increasing the atherosclerosis. This overexpression of pro-inflammatory cytokines are marker of chronic low-grade inflammation. Maintaining the balance of the pro and the anti-inflammatory responses of monocyte macrophages by the regulation of peroxisome proliferase activated receptor gamma, liver X receptor, matrix metalloproteinase, angiotensin, high mobility group box 1, receptor for advanced glycation end product, soluble protein 100, CXCL1, histamine, leukotriene receptor B4, interleukin 10, toll-like receptor 4 and scavenger receptor is the key way of preventive and therapeutic strategy for the atherosclerosis. Drugs or single compounds and their action mechanism of regulating these genes or proteins on the pro-inflammatory immune responses of macrophages in the atherosclerosis are illustrated here. Under chronic low-grade inflammation or pro-inflammatory immune responses, monocyte macrophages are so easily recruited and release cytokines that other immune cells became activated simultaneously to accelerate the immune response and production of pro-inflammatory proteins. Inflammatory proteins make monocyte form cells. Form cells inhibit cholesterol efflux. High-density lipoprotein produced by cholesterol efflux downregulate these immune responses. Ligand binding of toll-like receptor 4, so-called pattern recognition receptor or pathogen-associated molecular pattern, promotes the expression of target gene, production of pro-inflammatory cytokines, and consequently insulin resistance and atherosclerosis. There are certain kinds of toll-like receptors, saturated free fatty acid like stearic acid and oleic acid are important ligand of toll-like receptor 4. In contrast, free unsaturated fatty acid like omega-3 fatty acid suppresses TLR4 and consequently inhibit NF-kappa expression. Pipa gamma is a housekeeping gene present in almost all of the cells. Omega-3 fatty acid is a kind of pipa-gamma agonist. Pipa-gamma plays an important role in immune responses. H agonist transforms classically activated M1-type macrophages into alternatively activated M2-type macrophages. Ligand-dependent small-relation of pipa-gamma ligand binding domain, which target pipa-gamma 2 nuclear receptor co-repressor histone deacetylase 3 complexes on inflammatory gene promoters, prevent recruitment of the recapitulation 19S proteasome machinery that normally mediates the signal-dependent removal of co-repressor complexes required for gene activation. SUMO1 pipa-gamma and SUMO2/3 LXL inhibits distinct anchor clearance mechanism. LXL regulates innate immunity and inflammatory signaling in macrophage. LXL activation also reduces the inflammatory gene expression triggered by stimuli such as toll-like receptor ligand and cytokines by the mechanism of transrepression. Ligand-activated LXL blunts the induction of classical inflammatory genes such as inducible nitric oxide synthase, COX-2, MMP9, and various chemokines in response to LPS, TNF-α, and interleukin-1 beta stimuli. Toll-like receptor 4 activation of angiotensin II and receptor for advanced glycation and product activation of AGE suppressed by pipa-gamma. High-mobility glucox-1, a nuclear protein released passively by necrotic cells and actively by monocyte macrophages, and respond to exogenous and endogenous inflammatory stimuli, activates vascular endothelial cells and monocyte macrophages to express inflammatory cytokine, chemokines, and adhesion molecules. It was suggested that HMGB1 is a cytokine derived from both injured endothelium and activated macrophage to progress the atherosclerosis and other cardiovascular disease. Once released, HMGB1 stimulates neighboring endothelial cells to express various pro-inflammatory cytokines, chemokines, and adhesion molecules as well as RAG. This adhesion molecule enforces strong attachment of infiltrated monocyte macrophages to the endothelium, which should in turn further increase the release of HMGB1 and other cytokines. Receptor for advanced glycation end product detected on monocyte macrophage is a multiligand receptor of the immunoglobulin superfamily and involved in homeostasis, development, and inflammation. RAs interact with various ligands including HMGB1, advanced glycation end products, beta amyloids, and the soluble protein 100. HMGB1-induced activation of RAs was responsible for both NF-kappa B activation and neurite outgrowth by separate signaling pathways. Alternatively, RAs can signal through the activation of p38 MAP kinase and ORC1/2, finally leading to the phosphorylation and degradation of I-kappa B and NF-kappa B mediated activation of gene expression. Calgranulins, including soluble protein 100, A8, A9, and A12, members of soluble protein 100 family of calcium-binding proteins, also called myeloid-related protein 8, 14, and RAGE, respectively are associated acute chronic inflammatory disease. Angiotensin II upregulates the expression of amprin and inducer of MMPs. It is mediated by angiotensin I receptor, but not angiotensin II receptor. Inhibitor of NF-kappa B suppresses the effect of angiotensin II on amprin, so NF-kappa B is the critical factor involved in upregulation of amprin by angiotensin II. The activation of adenosine receptor A2A by adenosine blocks the NF-kappa B activation. An AT1 antagonist, CV11-1974, which is an active metabolite of candesartan, blocks the activation of PI3 kinase AKT pathway and suppresses the activation of MCP1 CCL2 system. Li-29-4004, an inhibitor of PI3 kinase, shows same activity. Losartan, an angiotensin II receptor antagonist, downregulates ADMA-stimulated NF-kappa B activity and expression in monocyte, and angiotensin II elicited loss generation and DNA binding activity of NF-kappa B in THP1 cells. Adexal agonist T0901317 downregulates the expression of MPO mRNA in human primary macrophage. Chiazolidine antidiabetics rosiglitazone blocks angiotensin II-induced toll-like receptor 4 expression and MPO secretion and downregulates the activities of the transcription factor AP1, STAT, and NF-kappa B. Anti-hypercholesterolemia drug atorvastatin downregulates C-reactive protein, serum amyloid A, TNF-alpha, interleukin-1 and 6 in human. Atorvastatin suppresses NF-kappa B activation and decreases the expression of mRNA of COX-2, MCP1, and MMP9 in OXLDL-treated human macrophages. Statins activate PPA-gamma and PPA-alpha through OC1-2 and P38 MAP kinase-dependent COX-2 expression in macrophages. Statin downregulates expression of mRNA of TNF-alpha or MCP1 induced by LPS. Statin decreases the secretion of MMP1, 3, and 9 in rabbit form cells. Fenofibrates downregulates angiotensin II-induced toll-like receptor 4 and MPO messenger RNA and protein expression in raw cells. Chiazolidine type antidiabetic pioglitazone suppresses PAM and LPS-induced toll-like receptor 2 and 4 expression in human monocyte and mice peritoneal macrophages and downregulates NF-kappa B P65 dependent DNA binding activity. Ezetimibe decreases the activity of NF-kappa B in peripheral blood leukocyte. Ezetimibe is a cholesterol-lowering drug. It decreases cholesterol absorption in small intestine. Quercetin-3-glucuronide, a major antioxidative metabolite of quercetin, is accumulated in the macrophages of the lesion site. Quercetin suppresses cytoplasmic translocation and release of high-mobility group box-1 in macrophage culture. Quercetin reduced HMGB1 or LPS-induced phosphorylation of P38 junk and OC in macrophages. GW647, a specific PPA-alpha agonist, reduces mRNA and protein expression of MMP12. Also, aspirin downregulates MMP2 and 9 expression. Interleukin-4 and 10 downregulates the expression and activation of MMP9. Acetyl-11-ketyl beta-phosphorylic acid, a NF-kappa B inhibitor, downregulates NF-kappa B dependent genes such as MCP1, MCP3, interleukin-1 beta, MIP2, VEGF, and tissue factor. Artemisinin reduces the expression and activation of amprin and MMP9 by suppressing the protein kinase C delta OC P38 cascade in PMA-induced macrophages. Avacitinib, fluvastatin, and PPA-alpha agonist downregulates mRNA expression and protein secretion of MMP9 and reduces activity of MMP9. Resveratrol inhibits amprin expression via both OC1-2 and P38 MAP kinase pathway in PMA-treated THP1 cells. ONO-AE3, 208, and EP4 antagonist suppresses COX-2-dependent ECM-induced MMP9 expression in low cells. EP4 is one of the prostaglandin E2 receptors. NS398, a selective COX-2 inhibitor, and aspirin reduces ECM-induced MMP9 secretion and sugar-glycolated elicited macrophages. 1,25-dihydroxy-vitamin D3 decreases MMP9 production in PMA-differentiated THP1 cells and human blood monocyte and downregulates mRNA and protein expression of toll-like receptor 2 and 4 in monocyte. PIL-284, a leukotriene B4 receptor antagonist, reduces the activity of MMP2 and 9 and stent arteries, and this downregulation of MMP9 activity was correlated with a decreased macrophage content in the lesion. U75302 and Li255283 downregulates phosphorylation of ORC1-2, JNK1-2, and AKT. This antagonist reduces LTB4-induced NF-kappa B activation and downregulates LTB4-induced interleukin-6, MCP1, and tumor necrotic factor alpha expression in human monocytes. SB20-3580, PD-098, O59, and caffeine-based ester reduce C-reactive protein-induced MMP9 activity.